Hello, my name is Jeff Mahr, and I teach biology at the Clarkson campus. And I want to set a timer here as I go. Actually, I need to look at the clock. And I want to give a little presentation about coronaviruses. And the first thing I want to mention about factuality of things is, it can be really hard actually to determine what is a fact and what is not. And there's actually, it's pretty rare that information is completely 100% factual, or at least there are cases where things can be sort of true in some senses and contexts and not others. And it can be very difficult, especially, when you're not at your expertise and you're not in your area. And so one thing I always consider whenever I'm getting information is I don't even worry about whether it's a fact or not. I consider the source I'm getting it from. And so, for instance, you can see here I'm laying out for you, like, I have some experience with human viruses, but not a single one of them is a coronavirus. So, I am clearly not an expert on coronaviruses, but I do have some expertise. So what I would do is, if I were listening to this, I learned something, interesting, or there was some sort of interesting tidbit, I might say, okay, I learned that from a community college teacher who, has some background in human viruses, not bad. But even experts can be wrong, and even people who don't even have a science background, it's totally possible for them to be communicating, complex information in a very nice way. So I would encourage you to just remember the background of the people you are learning information from, and consider that. Okay, so just to get something out of the way right away, the name of the virus is SARS CoV-2. The disease that it causes is COVID-19. There's some places where that might help a little bit to understand this talk and some of the news. Also want to get at a common misperception, or let's call it what it is. It's a conspiracy theory that I've heard frequently, and that is that this virus is somehow it's a biodefense plot by the Chinese government to disable the world economies, or keep us all in our houses or something. While there is Wuhan, China is where the first cases of the SARS-CoV-2 came up. There are some very large institutes there. It's a very large city, and there is the Institute for Virology, which has the very large National Biosafety Laboratory, in which they could make very dangerous viruses, and they might be making very dangerous viruses. But there's zero evidence for this being derived from humans making these. There are certain things we would look for that you would expect in a human-made virus. And I really won't be going into any of that because it's highly technical. I can sort of talk about it at the end if anybody's interested. But suffice it to say that there's zero evidence that this came from that. There are, however, multiple independent lines of evidence that this really is a bat virus that crossed over into humans. Now, as far as when did the current epidemic start, this is one of those things that it's sort of in that area of you can't know the facts. We may not ever know when the current epidemic started. So the first I became aware of it, and you could have become aware of it here in the United States, is a site, I don't know if you can see, is ProMed Mail. I would highly recommend if anybody's really interested in tracking global infectious disease. This is a site that is updated daily and is really approachable. And it literally is the first place you'll hear about infectious diseases. And in December 30th of last year, there was a patient posting. It was from a Chinese website. It looks like a government site. I think it looks like a finance site. And the translation of it basically boiled down to reporting these 4 patients with this odd disease they didn't know anything about. But the first sort of published, sort of peer-reviewed journal report of this was a case. It was published in February, but the actual paper was available long before that. And it was reporting 41 cases in Wuhan. And the first of those cases, the earliest case that one of these 41 had shown symptoms was December 1st. So that puts it back to, maybe, last week in November that this would have started. Interestingly, another thing that is kind of put about in the news media like fact and is not a fact, and that is that this started with the Wuhan fish market. There are many people involved in this first 41 cases that had no connection to the Wuhan fish market. And I would think to my eye, it seems very unlikely that the crossover event from bats occurred in the fish market. It certainly is possible, and we'll talk about the first SARS outbreak where that kind of thing did happen. But there's actually, a fair amount of evidence suggesting that the Wuhan fish market maybe had a role in amplifying the human infections, but that the crossover event for bats probably didn't happen there. But notice my questioning tone of voice there. The very first case that I've ever heard of, and I'm not even sure how to take this, so the South China Morning Post, a reporter published that she had seen Chinese government documents saying that the first case that they had was November 17th. So everything points to the crossover event, the zoonotic transfer, zoonotic infections, those infections that come from non-human animals into humans, occurring sometime in November, although we can't rule out that it happened somewhat earlier, since this is a virus where most of the infections are subclinical, meaning you don't go to the doctor for them because you don't even know you're infected. It's actually fairly rare that people get significant symptoms. And so it's entirely possible that this could have been rambling through the Chinese population for a while. Although we might never know the answer to when exactly the crossover event was. Okay, so I want to talk about the coronavirus as a family, because this is something that I see is often confused and is something that, normally when someone mistakes like virus for bacteria or calls like the flu virus as though there's only one flu virus. In any other time, that really wouldn't be that big a deal, and I just don't really come back and say anything. But we have to say something when, I don't know if you saw those memes early on where there was a picture of a Lysol can and it had the word coronavirus on it, and the person in the meme says, what the heck, I bought this last year. How did they know about coronavirus? And after I talking to a fair number of people, I get the sense that they really don't know what coronaviruses are. Coronaviruses are a family, and I'm going to tell you a little bit about the family history here of coronaviruses. So kind of interesting that the first coronavirus that humans became aware of is actually a chicken virus. So in chickens, if you farm chickens, there's a disease called infectious bronchitis, or another word for that is laryngotracheitis or gasping disease. And this is a disease that can kill 50, 70, 80% of a farmer's flock, which is really significant. And in 1933, this is a picture from a publication. It's a journal called Poultry Science, and in 1933, they, among other things, they determined that what was causing this disease was a virus. Okay, notice the word filterable there. That actually, if you know anything about how viruses were defined early on, you had to filter them to show they would go through a filter, but won't go into that. So anyway, they show that it is what is causing this disease is a virus. Now, at the time, it's really important to see that, yes, it can. So I can see some of the chats here. Yeah, so it depended probably on the flock. I imagine it depends on how you kept your chickens, right? If they're all packed together, then it would transmit quite easily versus, more free-range chickens. It's going to be... Anyway. So anyway, this was not known. So this is 1933, first coronavirus, but coronavirus is never used in this publication because coronavirus, the word, only came about in 1968, right? So at this point, when they published this in 1933, all they could say is it's a virus, right? We now know it's a coronavirus, but that's that's a term we only invented much later. Now, a nice way personally for me to think about the history of scientific discovery is to look at the papers, scientific papers that were published about it. So this is a graph, a bar graph, you can get from the National Library of Medicine, and you basically can search and get different publications, scientific publications. And if you just search coronavirus, it gives you all the papers it has tagged coronavirus, and it gives this graph. And it's basically just showing you for each year about how many publications there were with, about coronavirus. And notice there's a big surge here. We'll talk about that. There's another surge here. We'll talk about that. And then notice this year we're only, I think May papers are already in PubMed. So we're not even 6 months into the year, and we just have this huge surge of coronavirus papers. So that first isolation of a coronavirus, once again, they didn't know it was a coronavirus, is actually 1933. Notice that there is no 1933 here. Part of that is because, remember, they did not know that this was a coronavirus, right? And so in the National Library of Medicine, it's not flagged as coronavirus. What you have to do is actually look at papers that reference, so you look at studies that reference this study. So later people published and said, oh, in 1933, this publication about IBV in chickens, right? And eventually they discover, oh, this is a coronavirus, right? So you have to kind of do detective work to track it down. So at some point, though, the papers were being flagged as being coronavirus, right? So in 1946, transmissible gastroenteritis virus in pigs. And then in 1952, a rather important virus, murine hepatitis virus. These are all three coronaviruses, but once again, in 1952, they wouldn't have used the word coronavirus because that wasn't a thing back then. Right? Some people might be surprised to see hepatitis virus. And so different viruses in different species. So each species has its suite of viruses which infect it. And many times the different viruses can cause completely different symptoms and different syndromes in different animals. So this mouse hepatitis virus, the reason I'm talking about it so much is because my father actually passed away a long time ago from multiple sclerosis. And this disease, right here, mice infected with mouse hepatitis virus are an important model in studying multiple sclerosis, because these mice actually develop an odd neural syndrome that really does look very similar to multiple sclerosis, even though multiple sclerosis is not a viral disease. So a lot of times these discoveries where they're discovering something that has nothing directly to do with humans oftentimes blooms, and in fact, you can still find papers published, this last month that are building off of research on this virus right here. So anyway, need time to move on. This is my scientific fascination. So in 1968, the name coronavirus was established, and it's because when they looked at an electron micrograph, this is an electron microscope picture of a coronavirus. I don't actually know which one it is. It's probably either the chicken one or the mouse one. Those were the two most frequently studied ones. And he looked at this out here, and they said, well, wow, that really looks like the sun's corona. So maybe the astronomers can tell me what the corona is. It just looks like a patch of light reflecting back. I don't know how that works, but either way, this looks nothing like that to me. But they decided on the name, and now we have the name coronavirus. Superheated hydrogen gas, Jay Dunn says. Okay, so obviously, isn't it clear to you that this is superheated to hydrogen gas? Looks just like it. You're pointing at it, that was the corona. Yeah, sun's corona. I know that's the corona of the sun. I just don't know well, now I know it's superheated hydrogen gas. That's cool. Okay, so that's in 1968 that the family is named. And there's a whole zoo of these viruses. So for every single virus that you've heard of, in humans, there is an equivalent virus in other species, and often more than one, and they only usually infect that species. So for instance, this is neonatal calf diarrhea coronavirus. That is a coronavirus that only infects cows and obviously causes some problems in neonatal calves. But there's a coronavirus in pandas, dolphins, even beluga whales. And notice, I'm not showing you all of the coronaviruses that have ever been discovered. I just picked a few. And now back to the stuff that's relevant to our pandemic here. And that is the discovery in 1962. There was a publication of the first human coronavirus. Now, this study was kind of interesting because it was in England. It was in a boys' boarding school, where they lived at the school, and if the boys got a cold, they would take a little bit of their nasal secretions and try to get virus out of it. And lo and behold, they discovered the first human coronavirus. And this is just, I can't figure out from their paper. I'm still trying to figure out if it's 229E or OC43, though it's one of those two. But it turns out there's actually 4 human coronaviruses that are, I'm calling them mild. I think that might be kind of dangerous to call any virus mild these days. But I call them mild because they cause roughly about 20% of the common cold. So there's other viruses that and bacteria that cause colds, but about roughly 20% every year are caused by these. So they circulate in the human population. And you've had colds and you got better, I assume. Otherwise, you'd be dead and you wouldn't be listening to this WebEx. So the reason you got better is because you mounted an immune response to that. Every time you have been sick, you've gotten better because your immune system recognizes the pathogen and eliminates it. Well, there is something that is a little bit alarming related to these viruses and the immunity that relates to the current pandemic. And that is that unlike other viruses and bacteria, the immunity that you get to these 4 viruses that cause common colds, it doesn't last very long. After a couple years, it's gone and you can be reinfected. Now, this does not mean that the SARS CoV-2 virus will act like this. It's entirely, none of these are SARS viruses. So SARS is kind of its own little family. It is a beta coronavirus. But it's entirely possible that it won't act like this and we'll get a vaccine that will work completely. But this is something that true experts are kind of talking about when they're making a vaccine. They need to think about, is this possibly a vaccine where we'll need a boost maybe every 2 years or something because the immunity is waning? We don't know. It's just another complication that might pop up. All right, so now we get to the first SARS epidemic, and that happened when in 2002, December 12th, I was defending my dissertation at Emory University, and we were right in the middle of the SARS epidemic. I have no memory of it because, of course, I was busy being an expert in adenoviruses. I was aware of it, but I didn't know much about it. But in 2002, into 2003, there was a global epidemic, and it came out of southern China, and cases reached other countries. It did not have the same kind of global spread, reaching virtually every country like SARS CoV has. But, over 8,000 infections and a case fatality rate of about 10%. Case fatality rate is very important. It's something that you might have heard debated or talked about in terms of this current SARS CoV-2. And case fatality rate is a very simple fraction. It's just the number of people who die divided by the total number of people infected. And that tells you, sort of roughly how dangerous this whatever, virus is you're considering. Well, for SARS-1, the total number of infected was actually easy to determine. With SARS-1 in 2002, 2003, there was very little of this asymptomatic cases. It wasn't zero, but if you were infected with this, you knew about it. So this case fatality rate is actually very, very solid. Unlike the SARS CoV-2, the current pandemic, the case fatality rate you can hear people arguing about, and I honestly, ask me in 3 years what it is, and I'll give you a very confident answer. For right now, I'd say it's going to be probably less than somewhere between maybe 0.7% and 1% of cases dying. But that means nothing, because what you need to be thinking about is people who are in sensitive populations, older folks, people with diabetes, high blood pressure, kidney problems. For them, the case fatality rate is much higher, and I think it approaches 20% for some of those populations, and that's just nightmare. And I'd like to point out, 1% of a million is still quite a lot of people. So 1% or even 0.7% is quite high. So in the first SARS outbreak, this 2002-2003, the wet markets in China, where they sell quite a bit of wildlife and animals to cook, were definitely an issue. Unlike this current outbreak, where I'm not really certain it was an issue, in SARS-1 outbreak definitely was. This cute little thing here is a palm civet, and palm civets clearly spread the infection. There were the earliest cases were linked to wet markets, and it was palm civets that were positive for the virus. We know that the palm civets, it wasn't a palm civet coronavirus, because when they looked at wild palm civets, which are very rare from what I understand, but wild palm civets didn't have a coronavirus, but the ones in the market, some of them did, which probably tells you that palm civets were infected sort of as they went to market maybe, or at the market. But either way, when you look at the virus, it's a bat virus that infected a palm civet that then in the wet markets transitioned into humans. Okay, so I want to compare these two viruses. So SARS-CoV-1 is the virus that caused the first SARS outbreak. SARS-CoV-2 is the one that is currently spreading all over the world. So most of the transmission of SARS-1 happened in hospitals, right? So there's a difference. We make a distinction between transmission events that happen in a hospital, where you have to get very close to patients, right? Doctors and nurses have to get right up in the faces of their patients. That's different from community transmission. Community transmission is transmission that happens in, grocery stores and banks and buses and classrooms and things like that. SARS-1 had very, very, very little community transmission. It was mostly in hospitals. SARS-CoV-2, the current one, the problem is most of the spread is in the community, and it is also able to spread in hospitals as well. Another really big issue is when the transmission happens. So with SARS-1, transmission only happened once someone was very sick and showing symptoms. That's when they had the high enough levels of virus to actually transmit it to another person. So pretty easy to avoid someone if they're coughing up a lung, like in the store. You just maybe go down the spaghetti aisle when they've cleared that aisle. But with SARS-CoV-2, the whole problem, transmission happens, we know, before people show symptoms. And that means you can't know who's infected. And also, it means you can walk around and have it, not know it, and be spreading it. By the way, it looks, from the infectious disease true experts that I listen to, it looks more and more like there are very few truly asymptomatic cases with COVID-19. When they interview people who test positive, who aren't, quote unquote, aren't sick, these infectious disease doctors are saying when they really push the people, they find that they were in fact sick, but it might have been just diarrhea or something they thought was allergies or something like that. So it looks like there might be a lot less asymptomatic cases than we currently think. However, there's still what I would call subclinical. In other words, you have symptoms, but it's nothing that would cause you to think you have the virus. So anyway, just a nugget there. Anyway, the response, so this is 2002, right there, that bar there. 2003, there's a big increase, or at least an increase, in scientific papers about coronaviruses that kind of keeps going up. And I want to actually throw something in. That's an odd transition because I didn't intend to throw this in, but this is something that's super cool about the current outbreak. So what you're looking at is a diagram of the genes of SARS CoV-1. So this is the genes of the virus that caused the first SARS epidemic. And this are the genes of the SARS CoV-2, which is causing the current pandemic. Notice they look very similar. Each of these boxes basically is a gene. If you've had a Biology 1103 or 2107 or a good cell biology class, basically each one of these is transcribed and translated to make a protein or sometimes several proteins. But anyway, all right. Notice that SARS-CoV-1 and 2 are very similar. That is because they are kissing cousins to each other. All right, so interesting that during the SARS-1 outbreak, by the middle of the epidemic, what they were noticing was isolates of the virus. So they would get virus from patients that had these deletions in the what's called ORF8, this gene area right here. There were these deletions where this was just gone, and they were viruses that would continue to transmit. They weren't. It's not like they were at a disadvantage in spreading. And then even later, there were bigger deletions in this region. And once again, these viruses still spread. And notice that I've put here less dangerous, right? These are actually less dangerous. So this paper right here, Mahr et al., what they did in 2018 is they decided, let's fix these viruses, so these viruses with these deletions, and let's see, does that increase or decrease their ability to replicate? Because many people might think, oh, what's happening is the virus is getting more dangerous, right? And so if we restore the genes that have been deleted here, maybe it'll be less dangerous. Well, actually, the opposite is true. When they repaired these viruses and compared it to the deleted viruses, there was a 10-fold increase in the viral replication in cells when they infected human cells with these. So these deletions are actually compromising the ability of the virus to replicate. The virus looks like it's actually mutating to be less dangerous. And that actually makes some sense if you think about it, because okay, you're a virus, you're inside a human being. If you do not get inside another human being, you're going to be dead in the water. So you have two choices here. Do you want to be a virus that causes severe symptoms and the person's coughing up a lung and obviously sick? Or do you want to be a virus that's a little sneakier and causes less symptoms so that the person continues to, the person you're in continues to touch doorknobs and touch money and touch people and get close to people? So this actually fits with some evolutionary thinking. In addition, that idea that viruses will get worse, will mutate to get worse, there are no examples, there are none, where a virus, after it goes into the human population, mutates to get worse. But there are many examples from many different viruses where the opposite happens, where they actually have mutations that cause them to sort of get less dangerous or get better. Ooh, I'm running out of time here. Okay, so the reason I'm telling you this about it is very recently there's a publication showing that 4 different viruses have been isolated from 4 different people with deletions in the same region of the genome that the SARS-1 deletions were in. So the same thing seems to be happening that happened in SARS-1. We don't know where, what this means or anything, but it's very interesting. Okay, so I'm going to kind of move ahead quickly here to talk about the second SARS epidemic. And let's see, hints why we think they're becoming more. I'll tell you what, I'll answer the questions afterward. I got to skip through some slides to make sure I don't take over other people's time here. But so the second SARS epidemic it's actually called MERS, which stands for Middle East Respiratory Syndrome. And Middle East Respiratory Syndrome is the virus is called MERS-CoV. It's a camel virus, and the infections, the epidemics, plural, happen in Saudi Arabia, Oman, and the United Arab Emirates. And oftentimes, people will get on planes and travel to other countries and spread that. This is a disease where there's limited community transmission. Most of the transmission happens in hospital setting. There's some evidence for asymptomatic transmission, and there's some evidence that it might be increasing too. I need to really read a lot more on that to understand how I feel about that. But the MERS outbreak is still happening, right? And let's see where it is. Here we go. So this slide here, the camel virus actually came from bats. I'll skip over that. We know that the bat to camel infection from the bats actually happened before 1983 because there's blood samples from camels in freezers that they've tested. And this is something that is happening pretty much every year. So in 2012 in Saudi Arabia, there were a few cases, more in 2013, 2014, it really took off. And then notice every pretty regularly, there are these epidemics of this virus, this MERS virus in Saudi Arabia. And one thing that probably, hopefully people are thinking about is the potential that this virus could switch over, could mutate to be more transmissible in a community setting, because this would be quite bad. This has a high fatality rate as well. And let's see, what time do I have here? Okay, let me just talk really fast, rush through some slides. I hear my girlfriend actually asked me this, and it's been asked this by other people, this idea of can you be reinfected with SARS? So if you get SARS, there's people who get the SARS and then get better, and then later will test positive for the virus. So what's up with that? Are they getting reinfected? The short answer, no, they're not. So SARS CoV, this is the genome for it. The current test for SARS CoV that you get, the most frequent one, is called an RT-PCR test. And what they're literally testing for is a piece of the viral genes. And what they do is they essentially chemically amplify it, and until the point where they have billions of copies of that piece of the virus. And then they can do things like, DNA sequence it and stuff like that. So that is the current test, the RT-PCR test. What it's testing for is viral genes. Well, let me ask you a question. You could go right now into some of the classrooms on the Clarkston campus, and you could find my DNA in those rooms. Does that mean I am in the room? No, it does not, right? Obviously, we leave DNA behind us, and that is the same thing is true for the virus. So viruses fall apart and die. That's a whole nother issue. But because you find the virus, the viral genes, does not mean you have found infectious virus. And I'll end with this here, the most sort of hopeful thing, that a recent study has shown in rhesus macaques, an animal model, that they infect them and then they get better, and then they try to reinfect them with this virus and they cannot be reinfected. Now, this obviously isn't humans, but that is very hopeful because in many ways rhesus macaques do resemble the human immune system, their immune system does. So this really does suggest that protective immunity, at least within the time frame of a year or 2 years, is going to be doable. So anyway, I was extremely nerdy and put links to every statement I've made here and the papers if anybody is interested. If anybody has any questions that you don't want to ask now, just email me any questions at my GSU address. I'm happy to answer questions about the virus. And then also just a bit of self-promotion here. I have a podcast that I occasionally do virus episodes for on there. So anyway, thank you very much. And Ricky, what do I do now?
Well, thank you, Jeff. Hang on for a minute. I think there might be a number of questions. So if you have a question, well, yeah, first of all, great talk, great info. Yeah, if you have a question, while you're asking the question, go ahead and unmute yourself. But when you're done asking the question, please mute yourself again. So, we're waiting for questions. Well, I have one to get it started. Well, it turns out that at the end of January and into early February, there was actually something passing through my son's team, and going by the symptoms that we all had, it sounded pretty close to the coronavirus. But of course, officially, the virus hadn't been documented in the U.S. yet. But I don't know, I've been sort of hearing from, reading different news sources that it's entirely possible that the virus was here before the official first case was announced. So, first of all, my question is that a possibility? And then second, how long until the antibody tests are available so we can find out for sure?
Yes, the antibody test. So yeah, it's pretty clear to me that the virus was here before we acknowledged it officially. Exactly when, probably they could figure that out by looking at banked blood and going back and looking at that. I know there's at least 2 labs that are trying to do that to figure out what are the earliest blood samples that have it. And I've heard similar stories from other people that it's entirely possible that for some people this is already ripped through. But once again, you don't know. It was influenza season. There's lots of other things that affect you. The antibody test, I think the tests are currently out there. The question for me will be, okay, the kind of test that I can afford, is it a quality test? Because I've done those kind of tests in different contexts, and I know sometimes they're trash. And okay, so let's say I have an antibodies positive for it. Well, can I trust my test? Because I'm about to go walk out into a grocery store and potentially bring home, the virus. And my worst case is honestly my kid's mom and I, if we both get very ill. I'm not particularly concerned about this virus, but we're trying to protect each other. So if we get sick, it doesn't happen both at the same time, because then who'd parent our kids? Yeah. There's another question here. If the virus started out subclinical, how is it that it now isn't? Okay, so the subclinical part. So someone asked, if the virus started out subclinical, how is it that it now isn't? Okay, so that part of the question, we don't know that it started out subclinical. We know that what we know is a vast majority of infections are either so mild that the person really doesn't know they've been infected, or they get, fairly serious infection that they don't think they need to go to the doctor. But there are an important subpopulations that will get sick enough that they're at risk for dying. Think about if that had been circulating in China, how do we know? Like, let's say somebody dies, they get pneumonia, and then they go into some sort of organ failure. If the doctors don't think to take samples and to do the test to figure out why they died, you would never know why they died. This is why coroners are like the most important thing there is in cities, because if we don't know why people died, we can't make any progress, and it's not obvious why people die. Most people actually die of kidney failure, right? You can lose a lot of organs and compromise a lot of functions, but your kidneys, man, that's a knife's edge there. So it's possible that people were dying. I don't know that it's likely, but it's possible people might have died at a low level, and it took the virus getting to a certain point where enough people were infected and went to the hospital. But once again, we don't know that. Let's see, be tested, you have to be evaluated. Okay, yeah, so to be evaluated from questionnaire of similar symptoms went to the virus okay, so I don't understand the second part of this question, but I get from it that the symptoms of the virus, so symptoms of infections it's very difficult to tell from symptoms what you have. That's something that when I teach microbiology is a surprise to many students, that different diseases all look like flu-like symptoms. They all feel the same. And so you cannot diagnose based on, flu-like symptoms, because there's a billion things that cause that. So anyway.
Well, just to talk a little bit more about my case, it was kind of the weirdest flu-like symptoms I've ever had. Because while I was having it was just like really deep-seated, like in my lung, like a constant tickle, constant cough, and then for like a couple of days I was like totally low energy, and I actually felt woozy, as if I drank some alcohol, but I didn't. Yeah. So I mean, I don't know. Obviously, the jury is still out. Hopefully our healthcare providers will get an excellent antibody test going. And I was also surprised because, actually my husband came down with it first, and then it took a week for my son to get it. And then it took about another week for me to get it. So it wasn't like, the typical 3 days.
Yeah.
And of course, my husband has, like exercise-induced asthma or cold-induced asthma. I mean, he grew up with 2 parents who were chain smokers. So he actually took the longest to finally shake the cough. I think it took 3 to 5 weeks for him.
Yeah.
And then my daughter never really got any, and of course none of us ever got sick enough to actually go to the doctor and do a test.
Yeah. One other thing I just want to comment, somebody said bats need to go. So good luck getting rid of the bats. 25% of mammalian species are bats. So think about that for a second. Of all the mammalian species you've ever seen, if you were to put them all and make bar graphs of how many species, 25% of the species are bats. There's a lot of them. And they play really important roles. If you get rid of the bats, they're getting rid of the mosquitoes. So what do you want? Coronavirus, or do you want Zika virus? So, But I do understand.
Jeff, I was just kind of kidding.
Oh, okay. I'm also just, I understand the sentiment too. They creep me out a little bit, even though they're cool.
Well, I mean, plus we might as well get rid of people because they make the choice to eat bats. We don't have to eat bats. We have enough other food sources.
Well, so eating bats didn't necessarily have to happen here. So that's actually the least likely way it happened. The most likely thing is in many areas, bat guano in caves is harvested to use as fertilizer on fields.
Oh,
and
okay.
We actually didn't know about any coronaviruses in bats until after the SARS-1 outbreak. So the early SARS-1 outbreak, they were just like, where is this coming from? We don't know. And it wasn't, I think, until 2013, where they said, oh, wait, it's in bats. And they would go into these bat caves and find tons of just bat guano, and there was just aerosolized virus everywhere.
Wow.
So I think the more likely thing is this outbreak is farmer went into a field trying to harvest some bat guano, bat poop for non-biologists, and, that's how they got infected maybe, and that kind of went that way. So.
Yeah, well, it's kind of like the deer mice, right in the West. There's a respiratory thing going around with deer mice or field mice or something.
Yeah. So.
Well, are you receiving any more questions?
What was that?
Yeah, I have a question. In China, they released the ban or the quarantine after 76 days. Is that something that we could possibly be moving toward, or is there no way to really know when it'll be safe?
The short answer to your question is there's no way of knowing when it will be safe until we do it and determine whether or not it was safe or not. So I listened to about 3 different podcasts by nerdy virologists, and all 3 of them, a repeating theme is paying attention to what's happening in Wuhan right now, because that's the question is, did it go down in China because they were able to socially quarantine people and keep people isolated, and it went away? And when people start interacting again, well, the virus is still circulating. Will it increase again? And I honestly haven't looked into that since the weekend. But all of the experts are really paying attention to what's going on to Wuhan right now because of what you just said. They're releasing the quarantine. So, and that's something I worry about here when President Trump talks about opening up the economy. I certainly understand we really do need to try to figure out a way to get economic activity going, to get money circulating to people. People are losing their jobs. But I worry that what could happen is if suddenly people start getting together in big groups, you'll see surges happen. That's definitely something that could happen. If we were a totally rational society and we could control what people did, we would have combination of antibody tests and PCR tests to really figure out who can go back out and play in the grocery store and who could not. But of course, we're not rational, and we're not in a country where we've managed to do a good job of testing. So we're just going to have to, I don't know what's going to happen, honestly.
We also didn't do, we weren't as strict as China was with the social distancing. Like, for instance, in like grocery stores, they're still implementing a lot of the distancing. I mean, they have certain things set up, but I went into Walmart the other day, and like someone can easily walk by you closer than 6 feet. So I feel like that has a lot to do with it as well. In addition to that,
we're two totally different societies. China culturally is a culture where the good of the group overrides individual freedoms. Right? And there are certain ways in which that's good. There are certain ways in which that's bad. We are a society where, I'm an American and you're not going to tell me what to do. Even though there's very few people who would say it like that, I think we have a reaction to being controlled that does make this problematic. Keep in mind, when you walk past people in the store, for really a good chance of getting this virus, you really need to stand in front of someone quite close for more than just a passing moment. I mean, ideally, you'd like to be 6 feet away, but if you pass, a couple feet of someone, and it's a very brief pass, it's not a zero chance, but the chances of you being infected are not very good. It does take a bit more than that. Somebody commented here, some people have received this virus, recovered it, but found later to have received it again. What does this entail? So the last part of my slides that I skipped, so they did not truly recover. What they're finding is long after you recover, you continue to excrete the virus genes. So you essentially produce virus that there's no infectious virus that they're producing for weeks and weeks, but you can detect the RNA from the virus. And so all that is just the virus infection hasn't truly ended. It's just you're likely not infectious. So what they're finding is that the window when you're infected is when you're producing quite a bit of infectious virus in your nasopharyngeal cavities. If you don't have a certain level of that virus, you might be infected, you might be producing virus in your feces, but it's dead virus. So likely that's what's happening when they say they recover and when they receive it again. No, they didn't receive it again. It just means they tested positive again, and that's because people clearly continue to excrete the dead virus for a while.
Okay, but I mean, they wouldn't get sick again.
No.
Okay.
I'm aware of no cases published cases where that's happened and the person gets sick again. And that's an important little point there, because I don't believe anytime I read something in the news, even if it's legit and it agrees with what I think I know to be true, I will still, if it really matters, try to go find whatever that article is based on and try to look it up myself, because sometimes they get it wrong. And this is a good example. Like when you say, this person says, found later to have received it again. No, they never really got rid of it. And that might seem like a subtle point, but it's not. They haven't been reinfected. So they haven't truly gotten rid of it. So subtle things matter.